This video is to help you revise the cell and it's geared towards the Irish Leaving Cert Biology course. So the smallest unit of life is the cell. This basically means that the smallest living thing has to be made up of at least one cell. Take, for example, a bacterium. It's made up of one cell. Cells have three features. So they have cell membranes, they have genetic material, DNA, so deoxyribonucleic acid, and cells contain cytoplasm. So where do cells come from exactly? Well, cells have to come from other similar cells. So skin cells have to give rise to other new skin cells. And this is continuity of life. All cells come from other similar pre-existing cells. The microscope that you use in school is a light microscope. It can magnify by up to 400 times. It gives you a very basic view of the cell, the outline. You can see the nucleus and the cytoplasm. To see structures such as the organelles like mitochondria and chloroplasts, you would need an electron microscope. The question that you're often asked is to calculate the magnification, the total magnification of your scope. So you look at the eyepiece lens first. That usually says multiplied by 10, it's magnifying 10 times. Then you choose the objective lens that you're viewing through. So if you're using the high power lens, it will say multiply by 40 on it. So to calculate the total magnification, it's the eyepiece multiplied by 10 and the objective lens multiplied by 40, which gives you a total magnification of 400. So when we are looking at stains under the microscope, it's important that we get a good detailed picture. So stains are applied to the cells to make viewing easier, and you can see that the nucleus here takes up most of the stain. The cytoplasm does get stained too, but not as darkly as the nucleus. And this is a picture of cheek cells, and cheek cells are animal cells and they've been stained with methylene blue. We did also look at plant cells, onion cells to be specific, but the stain for onion cells was iodine. What you're viewing under your light microscope is the protoplasm. It's the living content of the cell. It's the content surrounded by the plasma membrane. So it's everything inside that plasma membrane. These are the two classic pictures of the animal cell and the plant cell that you are presented with as viewed under a light microscope. When the cell is viewed using an electron microscope, you're getting a very detailed picture of the cell. This is the ultrastructure of the cell, and this is the definition you need to know. It's the view of the cells you would see using an electron microscope. Electron microscopes can magnify by up to 1,000,000 times, so you're getting a very detailed structure of that cell. There are two types. There's a scanning electron microscope, which gives you a detailed three-dimensional picture of the outside of the cell, and then there's a transmission electron microscope which gives you a very detailed enhanced picture of the internal view of the cell. So when you do view your cells using your electron microscope, you get to view the organelles, these tiny little bundles that appear to be floating in the cytoplasm. They are cell components and they perform very specific functions. The two that feature the most frequently on your course are the mitochondrion on the left here, known as the powerhouse of the cell, because inside the mitochondrion, energy is going to be released from glucose molecules, and that energy is going to fuel every single reaction in an organism. In the chloroplast, this is unique to plant cells, and it's inside the chloroplast that photosynthesis takes place. The electron microscope has enabled us to see the detailed inside structure of cells, and we now know that cells are classified as being either prokaryotic or eukaryotic. So what exactly does that mean? Prokaryotic simply means that the cell does not have a membrane-bound nucleus and it does not have membrane-bound organelles. So that means that the DNA is not in a nucleus, it's not enclosed in a membrane-bound nucleus, it's in the centre of the cell, and there are no membrane-bound organelles such as mitochondria and chloroplasts. Then you have eukaryotic cells. These cells do have a true nucleus. They have a nucleus surrounded by a membrane, and they do have membrane-bound organelles. Examples would be animal, plant and fungal cells. They are all eukaryotic. So here you have a mushroom, that's a fungus, you have a daisy, which is a flower, and you have a caterpillar, which is an animal. Each of those organisms are made up of many eukaryotic cells. Both prokaryotic and eukaryotic cells contain these tiny organelles known as ribosomes. Ribosomes are not membrane bound, so they're not surrounded by a membrane, and that's why you find them in bacteria, bacteria being prokaryotic. The job or the function of ribosomes is to make proteins, and you'll actually find small ribosomes similar to the bacterium here in these two organelles, the mitochondria and the chloroplast. This is a picture of the ultrastructure of an animal cell. So let's go over some of the labels. You can see those little ribosomes that we were just talking about. You can see the mitochondrion, which is the powerhouse of the cell. You can see the cell membrane, but look at the nucleus. The nucleus is new now to you. The nucleus has a dark center, and that's called the nucleolus. And it's inside that nucleolus that the ribosomes, the little structures that make the proteins, that's where they're formed. Also, what you'll find in the nucleus is the DNA, and it looks like this stringy, noodle-like material called chromatin. So when we look at the ultrastructure of plant cell, you're meeting many of the same labels. So you have the mitochondria there, you have the nucleus, you have the dark center, the nucleolus, you have the ribosomes, you have the cell membrane, but you also have these unique features. So you have that cell wall on the outside of the cell membrane, you have those unique organelles that are called chloroplasts, and it's there that photosynthesis takes place, and you also have this large permanent vacuole, and it's filled with a liquid known as cell sap. This vacuole gives structure and support to the plant cell and is used also for storage. Plant cells are much stronger than animal cells, and it's because they have that cell wall. That cell wall is made of cellulose. Cellulose is a polysaccharide, and it's an important source of roughage and fibre in our diet. So now it's on to the structure of the cell membrane. The cell membrane has a very special structure. It's known as a phospholipid bilayer. So it's made up of two layers of these phospholipids. So let's take a look at a phospholipid. So a phospholipid has two parts. It has a hydrophilic head and it has a hydrophobic tail. The head likes water and the tail hates water. So phospholipids will always arrange themselves so that the heads are always touching water. That could be the outside of the cell or the inside of the cell, and the tails are always neatly tucked away. And this is why it forms a bilayer. Embedded in this phospholipid bilayer are special proteins known as integral proteins, and it's through these that large molecules, those too big to fit through the phospholipid bilayer, can pass to enter and leave the cell. For example, glucose. Other materials such as carbon dioxide, oxygen and water can just pass through the phospholipid bilayer. As well as the proteins, there are glycoproteins, and these are involved in cell signaling or recognition. All of those separate parts of the cell membrane, the phospholipid, the integral proteins, the glycoproteins, all of those, they can move around, and they do so. So for that reason, the cell membrane is described as a fluid mosaic model. It's made up of all of these little molecules that can move around and change position. One of the most important things that you have to know about the cell membrane is its function, and it has a number of functions, the first of which is that it acts as a selectively permeable barrier. This means it controls what enters and leaves the cell. So some molecules can move freely through the phospholipid, whereas others are tightly controlled. Two other functions or jobs that the cell membrane does is it plays a role in the recognition of molecules, so it's involved in recognizing molecules that touch it, and it also gives structure to the cell and separates the inside contents from the external environment, so it separates the inside of the cell from the outside. Often asked, where else in a cell would you find DNA apart from the nucleus? Well, in an animal cell you would find DNA in the mitochondrion as well, and in a plant cell you would find DNA in both the mitochondrion and in the chloroplast. Another question you're often asked is, where else would you encounter a membrane other than the cell membrane? And it has to be the nucleus. The nucleus has its own membrane called the nuclear membrane. Another question which you're often asked is to compare red blood cells to those cheek cells, those animal cells. Well, a red blood cell, known as an erythrocyte, has no nucleus and no mitochondria at maturity. These have been expelled. Red blood cells also contain many hemoglobin molecules, and cheek cells do not. That was cells. I hope the video helped. Remember, these videos never replace using a textbook, and they never replace your teacher's guidance. So make sure you're doing past papers and you're checking your answers. So as always, the professional looking black and white pictures are all from the Noun project. Although I am a pro member, I still wish to give credit.